Everyone. My name is Colette Gabriel, as Tom said, and I am the plant pathologist for the Plant Health Diagnostic Laboratory here within the Plant Pest Control Division at the Ohio Department of Ag. We deal with a lot of different samples, obviously. You may have heard from us before, you may have heard of us, but you probably have dealt mostly with your inspector. We are here in Reynoldsburg, Ohio. I'm not actually in charge, but I am the plant pathologist here. Dr. David McCann is the director here of our laboratory. And I'm going to, unfortunately, our internet's being a little weird, so I'm going to take myself off of video for now. And start. So there's a few different things that I'd like to go over, the aspects of what we do here in Reynoldsburg. The Plant Health Division here, we are really focused on trying to safeguard Ohio's agricultural and natural resources. We do this by performing exotic pest surveys. And then also, if we do find these exotic pests, we work on destructive eliminating them from the environment if possible. We deal with surveys for destructive insects and diseases on some aspects. We work with the different groups throughout the US to try and mitigate any issues that are coming here to Ohio or anything that's trying to leave Ohio and go to a different state. Here at the Plant Diagnostic Laboratory, we get all the confirmation samples. So if our inspectors don't quite understand something, we look at it and then confirm what it is. That's kind of what we do. We get samples from all over the state. Every once in a while, we also get samples that are coming in from a different state into Ohio, depending on whether or not it's been detained, coming from a different nursery, dealer, different vendors that sell plants from a different state to our state, just in case they have something that's not good, whether we were alerted previously or whether our inspectors found it. That's always a possibility. And we handle quite a lot of different types of samples. We'll go over that as well. What happens to these samples when they get here, and then what happens to the sample once we're done with it. So kind of just a quick recap. Our samples come from mainly our ODA inspectors. I'm sure a few of you have dealt with your ODA inspector as they're assigned to each nursery and dealer. We also get samples from our surveys. We have two major surveys that we deal with every year, and they're all through the USDA. The two that we deal with the most are our CAPS, which is mainly plant pests, so insects that are damaging the plant or bringing in disease to the plant, as well as our PPA, which used to be called Farm Bill. Those deal with plant diseases and insects. Another kind of smaller survey that we are partaking in, although we're not heavily involved, is with the ODNR, the Ohio Department of Natural Resources. They're doing an oak wilt survey, and we help them with samples from time to time. A major impact for our samples and our inspectors are nurseries and nursery stock dealers. These are two categories that we deal with in our programs. These programs help facilitate commerce by assisting shippers of plants and other commodities with various federal and state quarantines that restrict the movement of specific targeted pests and diseases. We provide different inspections for just general inspections to make sure things are clean in the nurseries and the dealer locations, as well as phytosanitary certificates for shipments of various different plant commodities that are destined for states and other countries. Usually what happens when, say, a new nursery shows up or there's a nursery that hasn't been inspected before, They're assigned an inspector, and that inspector kind of is the middleman between us and the nurseries. Once the inspector collects a sample, depending on whether or not they can figure it out on site, if they can't figure it out on site, it comes to us. And so once it gets here, it could go through various different tests or procedures before we finalize the report. Some of the more common ones that we see that are fairly easy to identify on a very quick turnaround table is boxwood blight, which is a very common nursery pathogen that's spreading through boxwoods. And then a few other like evergreen, needle blights, tip blights, things like that. If we can look at them under the microscope right away, they're fairly easy to identify as soon as they come in. If there's a situation where they're not as easy to identify, then sometimes we have to go look a little farther. You know, sometimes if we can't look under our scope and see it right away, we have to figure it out and determine what it is. And that sometimes involves further testing. A lot of things we do as well are insect IDs. These are fairly easy to do when there's distinguishable markers, but sometimes insects don't always have distinguishable markers. If it's something that we need to have further testing done for insects that don't have those markers, then we take those steps and identify them. If we can't identify something by distinguishable markers right away, and if we can't see something growing out of a sample as soon as it comes in, sometimes we have to grow those things out. And this could involve a simple procedure of a moist chamber where we place the sample into a plastic container with moisture and grow it out for a couple of days. That allows whatever is present to come out of the sample, and then we can look at it again under the microscope. If something is more in depth than that, where we can't just grow it in a moist chamber, we have to grow it out further. We do cultures. Sometimes it depends on what we're doing. We can use general media, which is very basic nutrients. And then we have culture media that is very specific, where there's a few different chemicals in them to allow specific organisms to grow. One of the big ones that we deal with, which is what we deal with ODNR, is oak wilt. As you can see in this picture, where these little dishes are here, these are our culture dishes. And these are culture dishes with oak wilt growing on them at various different stages and a few different media. So the one right here is a specific media, and this one is a more general media. So it just kind of goes to show how things look at different stages and how things look at different on different types of media, which is why we have to grow it in a lab and figure it out. And as an example for what happens with samples in our lab, we're going to kind of talk about oak wilt. Oak wilt is a pretty major vascular wilt disease. It's pretty prevalent within Ohio. It's in quite a few other states as well, and it spreads through injuries. And that injury can be from people, and it can also be from insects. The main vector for oak wilt is oak bark beetles and natidulid beetles. It's a very funny word. But then there's also us. But those beetles, they're attracted to wounds, which we potentially cause or storms cause. And then they take infected material with them, go to that tree, and then infect that newly injured tree. There hasn't been a whole lot of research whether or not tools can spread it, but it's always been a very good general practice to sterilize tools in between pruning and being around trees, whether you're pruning or weed whacking or mowing, things like that. You want to make sure you don't injure trees. These beetles, especially have certain time frames that they're active. And so we always recommend, hey, when you're dealing with these trees and there's a concern for oak wilt, that you don't prune during certain times and that you try not to injure the trees during certain times as well. This is just a photo I wanted to share. This is what they call a spore mat. The beetles are attracted to this, and then they take those spores and they spread them to healthy trees that have been injured. And this is just a general map of where it's been found. So it's a pretty broad location where it's been confirmed within the US. And there's other locations as well that may not have been on here. We just don't have record of them. Why is this a concern for nurseries? Because nurseries grow oak trees, as you probably know, but they also have oak trees in around their nurseries. Forest stands, whether or not you think your trees are going to be injured, there's always a high risk when you have a lot of oak trees around your nursery property, especially if those trees look like they're dying. They might have oak wilt. It just has a higher chance to potentially infect the product that's in the nurseries, especially if it's in ground versus in a ball or burlap. Although we deal with a lot of oak wilt samples, our oak wilt samples only come from ODNR. We don't deal with the general public, but there's another lab here in the state of Ohio that does also oak wilt samples, and that's the Ohio State University Plant and Pest Diagnostic Clinic. They deal with a lot more residential, they deal with a lot more commercial that's not regulatory related. Most of the oak wilt samples that we get from nurseries are samples that are required to be confirmed free of oak wilt. So for phytosanitary certificate for sending oak scions to another state, we would confirm that the tree that those oak scions are coming from are free of oak wilt. This is just kind of a general description of what a sample would look like coming to our lab. It would have to be a certain size. It would have to be a certain number of branches from a single tree. A lot of times when it comes to sampling, and our inspectors deal with this a lot, sometimes just any part of the tree doesn't work. You know, whether it's even for non-oak samples and non-oak wilt samples, you know, sometimes what's collected has to be what's actually causing the problem. So if you have a leaf, but the problem's actually in the roots, then that leaf doesn't show me anything. So this is just showing a description of, okay, so you're seeing some branches wilting. Those branches are what you would sample from, what the inspectors would sample from. That way, I'm actually looking at potentially the problem if we're looking for oak wilt. Just some more pictures of different various techniques. If you find, and this really doesn't work for nursery stock because nursery stock doesn't tend to be this large. But if it's nearby your nursery stock and you see something like this, then there's the concern that oak wilt's nearby. This is what our samples tend to look like. They're usually a half inch to an inch in diameter, and they're usually about 10 to 12 inches in length. Oak wilt's very unique in the sense that on red oaks, it's picture perfect 90% of the time. On white oaks, you probably wouldn't see normal symptoms that you would see on a red oak. And it's a very weird disease. And it doesn't always just affect oaks, it also affects chestnuts as well. If you look at these pictures here, you'll see this kind of spackling discoloration. That's very characteristic of oak wilt, but only in northern red oaks and the red oak family. Just some more pictures so you guys can see up close. This is another really key indicator, is that the vectors might be there, but they might not be transmitting oak wilt disease. If the oak wilt disease is not in the area, but the beetles are, it doesn't necessarily mean they're spreading it. So you might have branches that clearly have beetle activity. So these are little tiny holes. I don't know if you guys can see my mouse or not. Okay, good. So these are tiny little holes, and as you dig deeper into it, you would find galleries, and that would be the presence of the beetle. However, samples might be negative for oak wilt. So as I mentioned before, we tend to put oak wilt on 2 different types of media. This allows us to make sure that in case we miss it on the one media, it should grow on the other. There's 2 different types that we use. The one is a specific media, which is oak wilt media, and it has various different antibiotics and nutritional value that the oak wilt disease tends to like, but it grows a little differently than it would on our general acid potato dextrose agar media. The acid potato dextrose agar media tends to make things grow faster, and so it might end up getting overgrown by something else. This picture down here on the left is a really good example of a really clean culture. So nothing else is kind of outgrowing it, which is really good. But sometimes what ends up happening is we might get more than one thing growing on here. And if we aren't careful, the oak wilt disease growing on this plate might get overrun by something else that's not really important. Over here on the right, you'll see very distinct spore chains. These are kind of this is one of the main key identifiers for oak wilt. This structure is very unique, and a lot of the times, if you don't find these spores there, then we don't even think it's oak wilt. It could be something else, but this is one of the key identifiers. Oak wilt is also very tricky. Sometimes it takes 1 week for it to grow out. Sometimes it takes 3. It depends on the host. It depends on the actual disease itself. And there's really no rhyme or reason as to why there's different spans to when it grows, how fast it grows. It just does. This is just some more pictures of oak wilt media and the oak wilt disease growing. Here, this media is the APDA media, which is the acid potato dextrose agar media, and this is the oak wilt media, and these were plated at the exact same time. So you can kind of see how they grow differently. And this is just some more pictures of the spores and the fruiting structures. So just a couple things. There's a various different types of diseases out there that we can't identify by just looking at them right away. They require culturing and possibly even DNA extraction. These 2 are some major ones that are very similar to oak wilt in the sense that they're considered diebacks and wilts. And they're very difficult because they have very similar lookalikes and possible other suspects that they could be just by looking at them when you first get a sample. They're also spread by different beetles as well as potentially other mechanisms. The major one right now is laurel wilt. This is a really big one that's been moving up north. It was recently found in Kentucky near the Ohio River, and it's slowly moving up. And we're very worried that it's going to get into Ohio, and this could be very damaging to a lot of landscape and nursery stock. As you can see at the bottom, I listed out all the states that it's been currently confirmed in. So far, it hasn't been found in Ohio, but I know ODNR is doing survey Southern Ohio to kind of check and keep track whether or not it shows up. The other major disease that's been really popping up in our world, and definitely in the nursery world, is the vascular streak dieback. There hasn't been a whole lot of information about this disease as it's fairly new, but they're doing research mainly in Indiana and some other states. This disease has been found in nursery stock. I believe that's when it was initially discovered. And it's quite the variety of nursery stock. So we go from redbud to dogwood to spicebush to magnolia. And so it's quite a concern. This also has kind of a streak vascular discoloration that, you know, could be multiple different things. Initially, people thought that it was just a type of verticillium wilt, but they have been finding out that that's not the case. As research presents itself, once they find out information, there'll be a lot more out there to kind of help people understand what to do about this, how to control it, and how to protect their stock from getting infected. This is just a map to kind of see since 2020, well, as of 2020, what the map looks like for detections for Laura wilt. This is the one that's pretty close to us. We're not too happy about that, but we're definitely keeping an eye on it. All right, we are going to go back to this. So that's what happens when things need to grow out in our lab. It takes quite a few days, takes quite a few weeks. Sometimes, depending on the sample, it might have to be grown out and then also, you know, further testing. We can grow something out and then still be, okay, we're not 100% sure, so we have to confirm that. We also do a lot of different tests that involve nematodes. Nematodes are a real problem recently. I'm sure many of you have heard of the beech leaf disease. That's a big major one right now. They're still doing a lot of studies on that to figure out how it's moving, how it's getting into all the different beech trees. This is a huge deal for nursery stock, as a lot of places don't want this. And so if they find that the nursery stock has it, it can cause issues with shipment through other states, like to other states, as well as to Canada. So this has been causing a lot of news and a lot of issues. Nematodes can be in leaves, they can be in soil, not the beech leaf nematode, but just nematodes in general. And we find nematodes in various different types of samples. A lot of the times too, is it's not just the presence of a nematode. Nematodes can be either pathogenic or they can just be there. They might not be actually causing a problem. They might be feeding on bacteria or feeding on fungi or anything like that. So you have to make sure when we get a sample, we have to make sure that it's actually a pathogenic nematode before we actually report that in our report. And as mentioned before, sometimes testing isn't as easy as looking through a microscope or seeing certain structures. Sometimes we have to go a lot further. And I mentioned molecular work before, DNA testing, PCR. The major ones that we do, they're serological testing. This can be from various tests like amino strips, very similar to the COVID amino strip, where it gives us a control line and then it tells us whether it's positive or negative. Then we have what's very similar to the bottom left picture. This is our ELISA, and we do this test depending on what organism we're looking for. They're very specific. So it's not just something, oh, it's fungi or bacteria or virus. It's a specific organism that we're looking for. So it could be Phytophthora, which is a water mold, an oomycete. And we could be looking for just Phytophthora, and we'll run an ELISA test like this. This picture right here, this is a virus that we don't have any simple test to run. This requires molecular work. This is RNA extraction that then has to be sequenced to determine whether or not it's a specific virus. And if you're interested, this virus is tobacco rattle virus. So once these steps have been figured out, whether we've had to run DNA extraction or an ELISA whether they're based on surveys, whether they're based from nursery inspections or plants that have been taken because they've been shipped from another state into Ohio and they've been concerned, or we've been told that they might have something, we might end up having to send that lab out for other testing. So USDA Beltsville, they do a lot of confirmation of regulatory diseases and regulatory insects. So if we get a disease in our lab and we put it through all of our tests and it comes up that we believe, say, the big one is Phytophthora ramorum. If we get a sample that is confirmed to be Phytophthora ramorum in our lab, that gets sent off to USDA to get confirmed. And then once that's confirmed, then we have to go through the system. Our inspectors reach out to the nurseries that have this positive sample or the nursery that shipped in that positive sample, and we try to prevent that disease from spreading to other stock, from spreading into the landscape and becoming established. The other step that sometimes happens is sometimes we get a sample that every test that we have here that we can run comes up with nothing. And say we have a sample that is, we're certain it's a virus, but we just don't have a test for that specific virus. There might be another lab that we talk to, an external lab that can actually run that test for us. So that sample can get sent off to them and they run that sample for us, send us their information, and then we add that to our report. A few different examples are sometimes we can't carry every single virus test, especially with the ELISA tests and immunostrips, those all expire. And so if we don't have something in-house, then we can send it off to another facility that actually has that test. They've been using it and it's not expired. One of the major questions that we get is, where do these results go? Who do they go to? Since we have our inspectors, our information that we send out goes to those inspectors. Our inspectors are kind of like our first line of defense. They're that middle person that is the spokesperson for us to you, to the nurseries, to the dealers, and then they're the spokesperson to us for the nurseries and dealers. And so all that communication is run through them. Any type of questions that we might have, we ask our inspectors, then they ask the nurseries or the dealers. So whenever we write up a report, it goes to them. And then they then give that report to the nursery. They might even relay information like what might need to be done, the measures that might need to be taken, stuff like that. Another thing that we do try to do, the lab partakes in this as well as other various parts of our section here at ODA. We try to have as much outreach as we can. We want to be a resource to nurseries and dealers as well to the general public, to a point where we're letting them know what's going on. We want to make sure they're aware of what's happening in our state, and we work really closely with OSU to make sure that information gets out there and the correct information gets out there as well. You'll find that there's a lot of information out there on Google about different things. Sometimes they're not always the most reliable. You want to make sure that whatever you're reading is from a reliable source. And so we try to help push those reliable sources out there for people to make sure that they know this is a good source. This is information that has research backing it. This is the data that's been collected, stuff like that. Our website is, for the most part, fairly user-friendly. But there's always the opportunity, if there's any questions or concerns, we have different reporting tools and feedback options there that you can ask questions like, what about this, what about that, available to you. Another question that we get is samples that don't fall into the categories that I've already mentioned, that are from our nursery inspectors, that are considered for our phytosanitary certificates and regulatory surveys, regulatory pathogens and such like that. All the samples that are considered non-inspection, non-survey, non-regulatory samples, we don't take those. That's kind of why we have the inspectors in between. They're kind of the ones that make sure samples that come to us are the ones that we handle. Anything that's not a sample that we see, we recommend going to a different lab. OSU has a good clinic. They're up there in Worcester now. They've moved, and so there was a transition period when they weren't taking in samples, but now they are. There's also various different labs throughout the US that take samples like that. One of the big organizations that we deal with on a daily basis is the National Plant Diagnostic Network. It's a really good resource tool for not only diagnosticians like myself, but also for the general public. They help make sure people know about all the labs here in the US, neighboring states, different regions, and they also help those labs make sure they have the up-to-date protocols and make sure that they're doing diagnostics that are reliable. And that's it.
So, and I'll throw it over to you first. Is there anybody in your room that has questions? I had a couple that came up as Colette was talking, but Ann, anybody have a specific question for Colette?
Not at the moment. But
So
I have a
Colette-
question, which is, what is a regulatory pest or pathogen?
That is a good question. So we base all of our regulatory diseases and pests based on what's considered regulatory to the feds. So USDA, they have a lot of lists out there that list out all the concerned pests and pathogens, things that we don't want in the state of Ohio that might be in other states that are considered regulatory. One of the main examples is Phytophthora amorum. The other main example would be, for pesticide, would be box tree moth. Trying to think of some other ones out there. We deal with Ralstonia in various different nurseries and production sites. The other one would be tomato brown rugose virus. That's also a big one. Those would be considered regulatory, things that we don't really want into the country, things we don't want into our state. Regulatory means it's not here for the most part. Sometimes it's here and we're trying to eradicate it. If that answers your question, hopefully.
Thank you.
So Colette, you mentioned two initials, CAPS and PPA. What's the difference between those two things?
The main difference in my involvement is the funding and what pests we're looking at. CAPS tends to be a lot more on the crop commodity side. So we see soybeans, we see corn, we see oats, wheat, things like that. We also see some oat commodities in there as well, but it's mainly insect pests. So we're doing a lot of trapping, various different traps. We have bucket traps, wing traps, delta traps, and they're all different looking, and they all go out into different locations that we track those sites, and then we check them periodically throughout the year. And then we look at those traps and we check them to make sure that the insect that we're looking for is not there. And if it is there, then we have to hopefully take measures to get rid of that insect. PPA, it tends to be a lot more, at least on my involvement, it's been a lot more nursery based. So we do a lot of surveys with first sudden oak death, Phytophthora amorum, which is what it's called, where we do soil sampling in nurseries, doing soil sampling at garden centers, leaf sampling, things like that. We also keep an eye out for any samples that are coming in, like plant stock coming in from another state that's positive. The other ones that we do for those are phytoplasmas in orchards, vineyards, samples like that. So that has a larger range, but it's more of the industry that's selling things. That's, you know, commodities like wine and food and like plants, selling plants in general. Whereas CAPS tends to be in a field, crops in the forest. And I see there's a question between DNA and PCR. DNA, everything's got DNA and RNA. And so sometimes we have to extract those. PCR tends to always be there for the most part. What these are is when we extract DNA, it's only little parts of things. It's parts of the DNA. It's never an entire genome of that, say, the disease or insect. So we have to replicate those to be able to figure out what it is. And PCR is a way to do that for us. I could go into more detail if you would like. Or
it was more like, so you said that, so we use like test strips for, and you said that sometimes you don't have those. Is that you're looking, is that more PCR related or is that DNA, RNA related?
the test strips,
Yeah.
those are serological related. So when we use a test strip, unless it's something for our surveys, it doesn't go past that test strip. It's either positive or negative. So the test strip, very much like a COVID strip, those amino strips that you would get for a rapid detection, that's what they are. They're a rapid detection. So we can take those on site, grind up samples, put that test strip in a little baggie of solution, and then we can tell whether or not within 20 to 30 minutes that it's positive or negative for a specific disease, pathogen, things like that. So we might have a test strip for, let's see, tobacco mosaic virus. And we're looking for that. We want to make sure this plant doesn't have it. So we'll test it on the amino strip and it will tell us right away. DNA extraction and PCR, those are a lot more time consuming and a lot more materials involved in that. So it's something that's more specific. So we can tell whether or not something is an organism to species. Whereas sometimes with the amino strips, it can it's more general. It says it's it could be TMV or some other virus or other disease, but sometimes those amino strips will also pick up other organisms. So we can only be more general with them. Whereas DNA extraction will tell us the genus and the species when we take it through PCR and then sequencing. So DNA, PCR are kind of hand in hand. It would be really difficult to do DNA extraction without also having a PCR run and then also potentially sequencing or having a PCR run called real-time PCR, where we can use chemicals and certain techniques to determine whether that plant sample has a specific disease based on those reagents and primers and probes that we have developed for it. So it gets really into the nitty-gritty and details, and it's a really intense process. Usually it takes about a couple of days for those, but getting the samples and grinding them up and getting them ready and making sure that you have the right piece of the sample for that test can always be a little difficult sometimes, without the, knowing what you're doing and stuff like that. So it's quite the process. And we only do it for specific things here in our lab, mainly for our surveys. So we do phytoplasmas, we do sudden oak death. Those always go through PCR. And sometimes if we get a sample that we're really concerned about, say like laurel wilt or vascular dieback, streak dieback, we'll do PCR with that as well.
Did you want to just, because you're kind of there now, but did you want to mention the value of doing controls? Because I know you're big on that, kind of making sure that you have controls in place along with actually testing for positives.
Yes, so pretty much most of our tests all have controls. The only things that don't have controls are when we're just looking at a microscope and looking at spores and fruiting bodies and things like that. But any of the tests that we run, so whether it's a serological test, a molecular test, we have controls to make sure that that test is running properly. If that control isn't present or if it doesn't work correctly, then that test is invalid. And so anytime we have tests coming into our lab, like we're running in our lab, we always make sure that those controls are working properly and are present. anytime there's a situation where something's not looking right, you never ever want to be like, oh, it's positive, even though my control isn't working, so my test isn't running properly. So it's always a scary thing. You want to make sure those controls are working correctly. And that's why I mentioned, if we don't have a test, or say we had a test but it's expired and we had to get rid of it, we wouldn't keep that test, because once it's expired, there's no longer a guarantee that it's going to work. And then we're wasting that sample trying to run it on a test that's expired and it doesn't work. So we don't ever deal with anything that's expired here in our lab. Those controls are pretty important, having that in place.
Jacob, anything? You dazzled them with all your scientific talk, Colette, at the end. You see that? Will, anything?
Oh,
Anything
sorry,
from
Tom.
you, Will, over Willoway, or?
Nope.
Colette, sometimes you'll get a sample, like, let's say, for example, Japanese maple scale. Can you determine whether the scale is already dead or still alive, or is that something you can determine in the lab?
Depending on the stage, yes.
Okay.
So, if it's already gone into dormancy, it'd be a lot harder to tell whether it's alive or not. Usually you can tell based on how, and this sounds really silly, but based on how squishy it is. But the best time to tell if they're still alive is if the crawlers are still moving. That's one of the easiest indicators that they're still living. We'll get samples throughout the growing season, depending on when they're collected, like, hey, are they still alive? And they'll just be crawling with crawlers. And it's like, yep, they're most definitely still alive. And then other times it's like, okay, there's no crawlers, so we gotta check these, make sure they're not dried out. Because if they're dried out, then they're dead. They're not there. So it just depends on the time of year. One question is, how many samples do we process during the growing season? And this is very fluid. It can range between, and I mean this with not including our survey samples, it could range between 80 samples to 400 samples, maybe even higher. It just depends on the year. Some years are slower than other years, but we always have quite several thousand with our surveys. James asks if when you sequence a specimen, is this run through ODA database? There's a larger database that we go through. There's a few different ones, but the main one that we use is NBCI, I believe it's called. This is managed by a different outsourced, kind of an external group, not by us. And so all the DNA that's on there is usually published work. And so we compare our sequence to that database to allow us to determine what it is. And this helps us because, one, we can't have a database for everything in the world. So we try and make sure that we're comparing it to more than just what we get here in the lab. We do keep a database of whatever we find here, but it's very minimal and it's very short sequences. And so comparing DNA with what we get here in the lab doesn't make a whole lot of sense because it's not the full genome. So the database that we deal with, they tend to be full genomes, or at least larger parts of the DNA than what we currently have. Does that make sense?
Yep, thanks. That answers my question.
All right, any other questions for the group? We're good here. Colette, I guess I would be remiss to say that we do appreciate your work. I know that you take a lot of pride in carrying your work, and the fact that you have inspectors that rely on you, and then you communicate that back to the nurseries. We appreciate that. It's a good thing for all involved. So we appreciate your work.
Anne did ask a question about, have you examined any samples for vascular streak dieback? We have not received any samples in the last few years. A while ago, before they determined what this was, we did get a spice bush sample that we were concerned that we couldn't quite figure out. At the time, we didn't know anything about vascular streak dieback, and we didn't have anything to primers or probes to do DNA extraction for that specifically. It was sent off to another lab at one point, and I don't know if that was something they ever figured out or determined for us. But we have not actually had any samples since it's been determined and there's been information about it for us. Most of those samples, I believe, end up going to some of the universities. There's a lot of research still going on, so anytime we've heard of anything, they've usually been sending off to them.